Greetings everyone. For today's topic, let's discuss about the structure of viruses. When we say virus, it is a microscopic infectious agent capable of infecting living cells and hijacking their cellular machinery to replicate and produce more viruses. They are much smaller and simpler than single-celled organisms like bacteria, and they do not meet most of the criteria to call something alive. One of those criteria is to perform metabolism, right? And viruses didn't meet this. They cannot make energy from food, and they cannot reproduce out of their own capacity. Viruses need ribosomes, enzymes, and other crucial cellular components. And in order for them to reproduce, they need a cell to do it for them. Thereby, hijacking the cellular machinery was their way to produce copies of viruses. The structure of the virus comes in a number of forms. Some are rod-shaped or helical, like the tobacco mosaic virus. Some are icosahedral, like an adenovirus. Some have a membranous envelope covered with spikes, like the influenza virus, and the bacteriophage, a combination of rod-shaped and the icosahedral virus with some fiber tails. All of them carry their own genetic material. It can be DNA or RNA, containing the instructions for viral replications and function. A capsid, which is a protein coat that encloses and protects the genetic material, which comes in different shapes for different viruses and is made of smaller subunits known as capsomeres. Envelope. In some viruses, which have an outer lipid membrane envelope derived from the host cell membrane, and this envelope may contain viral proteins and glycoproteins, surface proteins, or spikes. These structures protrude from the viral envelope and are involved in attachment to host cells during infection. Matrix proteins in envelope of viruses which connect the nucleic acid to the capsid or envelope, aiding in the structure and stability of the virus, and viral proteins that, apart from structural components, viruses may carry enzymes and proteins necessary for their replication and infection process. Some examples of viruses according to the sizes are: first, the poliovirus that measures approximately 30 nanometers in diameter, causing polio in humans. The hepatitis B virus, roughly 42 nanometer in diameter, responsible for causing hepatitis B. The influenza virus, which is around 80 to 100 nanometer in diameter, which is responsible for seasonal flu outbreaks. HIV, or the immunodeficiency virus, which is roughly 100 to 120 nanometer in size, leading to acquired immunodeficiency syndrome or AIDS. And the last one, the bacteriophage, the size ranges from about 20 nanometer to over 200 nanometer in diameter. Let's move forward about how viruses replicate. As what I have said a while ago, viruses replicate by hijacking the cellular machinery of a host organism. And this process typically involves several key steps, the first of which is the attachment. The virus attaches to specific receptors on the surface of the host cell, often facilitated by surface proteins or spikes on the virus that recognize and bind to complementary receptor on the cell membrane. Here, certain viruses are able to infect certain kinds of cells, and this is due to the system of recognition that must occur between the two. In order to get inside the cell, a virus must be recognized by surface receptors on the cell. So there must be some specificity for this receptor to that particular virion. After the attachment, here comes the entry. The virus enters the host cell through various mechanisms, such as direct fusion with the cell membrane, endocytosis, which means engulfment by the cell, or injection of genetic material into the host cell. So, after the recognition occurs, the virus either injects its genetic material into the cell, if it is a bacteriophage, or the virus can be brought inside the cell completely intact through endocytosis. It is followed by the uncoating. Once inside the host cell, the virus shades its protein coat or the capsid to release its genetic material. It's either DNA or RNA into the cell's cytoplasm. So once inside, the virus disassembles and the viral DNA gets transcribed and translated by all parts of the cell that are typically busy working for the cell itself. The fourth one, that would be the replication, where the viral genetic materials takes control of the host cell's machinery, directing it to produce viral components such as viral RNA or DNA and viral proteins. These components are assembled into new virus particles in replication. Once there are many copies of the viral DNA, the capsid proteins reassemble and form new viral particles, up to hundreds or even thousands of them, which then exit the cell. It is followed by the assembly. Newly formed viral genetic material and proteins are assembled to create new virus particles. Once assembly and maturation are complete, the newly formed virions are ready to release from the host cell. The release mechanisms vary. It can involve cell lysis, or the rupture of the host cell, common in non-enveloped viruses, or budding, where enveloped viruses acquire a host-derived membrane and are released without causing immediate cell death. The released viruses then can go to infect other cells and continue the replication cycle. Sometimes, this process can damage or destroy the host cell. So let's look at the different mechanisms a viral replication can utilize for bacteriophages. The lytic cycle. With the lytic cycle, the host cell is terminated at the end of the replicative cycle. This happens once many viruses have been generated and the cell bursts open or lyses, releasing them to then go and infect other cells. Given the exponential nature of this process, just a few successive lytic cycles can destroy an entire bacterial population in a couple of hours. By contrast with the lysogenic cell, the host cell is not destroyed. This is because, rather than usurping the cellular machinery to exclusively produce viruses, the viral DNA is incorporated into the genome of the cell. We can refer to this kind of viral DNA as a prophage. This DNA remains largely silent, and the cell is able to divide many times, with each daughter cell also containing the prophage. Then some environmental signal may trigger a switch from the lysogenic mode to the lytic mode, where the prophage returns to the form of a separate cellular DNA molecule, and all of the infected cells could lyse at once. Apart from bacteriophages, other viruses have envelopes, which allow them to enter and exit the cell by endocytosis and exocytosis without destroying the cell. So it is important to the virus that it will be recognized by these surface receptors.